This is Unit 8A. Okay, pregnancy is the period from fertilization until the fetus is born. The gestation period is from the last. The gestational period is measured from the last menstrual period until birth, and the developing offspring. The developing embryo and fetus are called the conceptus. So from the very start, from the point of fertilization, right up until the birth, it's called a conceptus as a general term. And then there are more specific terms. Pre-embryo, if the conceptus is 2 weeks up to 2 weeks old, embryo from 3 to 8 weeks, and a fetus from 9 to 40, and it then gets delivered at 40 and is a newborn. So here you see the different stages of the conceptus. Pre-embryo in here, and then pre-embryo here, and then embryo up till 8 weeks, and then fetus until not just 12 but all the way to 40 and birth. Okay, now fertilization will occur when the egg and sperm meet and fuse to form a zygote. And in terms of the egg or oocyte, it's viable for 12 to 24 hours after ovulation, which normally occurs at day 14 of the ovarian cycle. So at day 14, the egg or oocyte is released and moves towards and into the uterine tube. Excuse me. Meanwhile, sperm that have been deposited in the vagina are viable for 24 to 72 hours. So for fertilization to occur, sex has to occur no more than 3 days before ovulation or 1 day after ovulation. So that's a peak period for fertility. 3 days, and optimum for a woman to get pregnant is to have sex between 3 days before ovulation and 1 day after. And that's the most likely or highest level of fertility period. Okay, now sperm that are released during ejaculation, just a second. Sperm that get released during ejaculation will either leak out from the vagina immediately after being deposited there, or they get destroyed by the acidity of the vagina environment, or they fail to make it into the cervix and then into the uterus. They could also be dispersed and destroyed, dispersed in the uterine cavity, never make it into the uterine tubes, and they can be destroyed by phagocytic white blood cells. And so only a minority will reach the uterine tubes where fertilization of an egg normally occurs. Now, sperm have to undergo some basic changes, physiological changes that are called capacitation before they can fertilize or before they can penetrate and fertilize the egg or oocyte. They have to become fertilization competent, and that capacitation, that ability to fertilize, that transformation to being able to fertilize, will occur in the female reproductive tract after ejaculation of the sperm in. So sperm that are produced initially don't have the capacity. They have to undergo this alteration. And the changes include hyperactivation, so that they can swim faster, and the tail movements are more forceful and erratic. And they have to undergo the acrosomal reaction. Remember, the acrosome is on top, right at the tip on top of the head of the sperm cell, and it's where there are enzymes, hydrolytic enzymes, that help penetration of the egg. So the acrosomal reaction is release of enzymes enabling penetration of the egg. Now that reaction, as I said, is shown in the diagram here, right there. That's the acrosomal reaction. So release, activation and release of the enzymes. And the reason that those enzymes are so important is that the egg or oocyte is surrounded by a layer of cells called the corona radiata. This is shown here in the diagram. And also a cap surrounding, not cap, it's a layer that surrounds the entire oocyte, and that's called the zona pellucida, shown here. Just below the corona radiata is the zona pellucida. And so in order for sperm to enter, they will bind to the zona pellucida, and they undergo that acrosomal reaction, releasing enzymes right there near the zona pellucida, and that allows them to penetrate in because the enzymes will digest the zona pellucida. It is not only one sperm cell that will undergo that acrosomal reaction. There are hundreds that are releasing the enzymes, allowing them to digest the zona pellucida and try and enter in. So once a sperm penetrates in and makes contact with the cell membrane of the oocyte, that involves a protein on the sperm called beta protein, which can bind to particular receptors on that egg or oocyte membrane, and alpha proteins, which allow it to insert right into the membrane. And so here again you see the corona radiata cells, the cells of the corona radiata layer, and just below that the zona pellucida, and the sperm cell approaching, undergoing acrosomal reaction, digesting its way through the zona pellucida and entering here into the oocyte and binding to the oocyte cell membrane. The cell membrane of the oocyte is right here. Okay, now what prevents more than one sperm from fertilizing the egg, which is a process called polyspermy? That has to be prevented. Only one sperm can penetrate in and fertilize the egg so that it becomes 2N, diploid. Remember, the sperm is haploid and the oocyte is haploid, and when fertilization occurs, it then has 2 copies of each of the 23 chromosomes. So that's the number you need, not more. So polyspermy has to be prevented. And polyspermy is prevented by 2 mechanisms. One is the fast block and the other is the slow block. And the fast block is a depolarization that occurs as soon as the first sperm to successfully enter and fertilize triggers a membrane depolarization in the oocyte, preventing any further sperm from binding and fusing with the egg or oocyte membrane. The other one is a slow block, and that's mediated by what are called ZIPs, or zonal inhibiting proteins. So there's an inhibiting protein which can destroy the sperm receptors on the oocyte membrane. Those are the things that the I can't go back, but those are the things that the sperm proteins will bind to. And in fact, any additional sperm that have managed to bind at that point but haven't entered, so one enters and triggers this fast block and slow block. Any sperm that happen to be already bound will detach because the sperm receptors that they're bound to on the oocyte membrane get destroyed. So they detach. And this prevents multiple sperm from fertilizing the egg. Okay. Remember that, and we went through this in the previous unit 6, where we talked about oogenesis. Remember that at the time of fertilization, entry of sperm into the egg, and the egg or oocyte is at that point called a secondary oocyte. That, and it's still undergoing meiosis, that will trigger the completion of meiosis of the secondary oocyte, that entry of the sperm, and you get the egg and a second polar body forming. So at that point, the egg or ovum nucleus will swell, and the two nuclei inside it, the ovum nucleus and the sperm nucleus, will approach each other. These nuclei at this point that are now both inside that egg, the ovum nucleus and the sperm nucleus, are called pronuclei. And then fertilization occurs when the two fuse together. The two pronuclei fuse together to form a fertilized egg or zygote. And here you see that process that I was talking about with sperm entry through the corona radiata and the zona pellucida, penetrating in the egg or oocyte, and it's a secondary oocyte, will release a polar body and will split into the egg and a polar body, or oocyte and polar body. And the sperm pronucleus is now, I'm going to change color, the sperm pronucleus is now inside the ovum or egg. The ovum nucleus is there, and the two are called pronuclei, male and female pronuclei, or male pronucleus and female pronucleus, and the two fuse to form a zygote. Once that fusion occurs, it's a zygote, a fertilized egg. So now you start getting very active mitosis, and you get division of the zygote, and it quickly undergoes that process, and cells are dividing rapidly at this point. And that rapid mitosis, division of cells, is called cleavage. And the very first cleavage produces two daughter cells called blastomeres. So when the zygote or fertilized egg is at the two cell stage, those two cells are called blastomeres, and that's in the first 36 hours that occurs. The blastomeres will, the two blastomere daughter cells form. After another 36 hours, so now we're at 72 hours, what you have is that the zygote has about 16 cells, maybe a little more, and it's called a morula. The whole thing is called a morula. And then by the 4th or 5th day, it's called a blastocyst. So the blastomeres are the first two cells that form by 36 hours after fertilization of the oocyte, and that's occurring in the uterine tubes. And then the morula is at 72 hours, and the fertilized egg or zygote is still in the uterine tube, moving towards the uterus. And then it's in the uterus by 4 or 5 days, and it's called a blastocyst, and has about 100 or more cells, and it's free floating inside the uterus. What has to happen at this point, well, I'll describe this slide describes the blastocyst. It has a shape that is like a hollow sphere, and it's filled with fluid. And there's a single layer of cells on the rim of that sphere, and they are called trophoblasts. But it also has a mass of multiple cells inside, an inner cell mass, and that is going to become eventually the embryo. The trophoblasts, that outer single layer, takes part in the formation of the placenta, and the inner part, which is called an embryoblast, becomes the embryonic disc and the embryo. So here you see the fertilized egg cell, initially divides into two cells, which are blastomeres, and then goes into further divisions, and then becomes a morula, and has I think 36 cells or more, and then enters into the uterus and is at this point a blastocyst, 100 cells or more, and will implant at around day 6 into the wall. Before that, it is free floating. Here it's implanting into the wall. Before that, it was free floating in the uterus. And as I said before, the structure has a fluid-filled sphere shape, with the outer part or outer layer of that sphere being the trophoblast cells, and an inner cell mass, which will become the embryonic disc and then the embryo. Okay, the process of implantation starts at about 6 to 7 days after ovulation, which was day 14, right? So day 14, the egg gets released, moves into the uterine tubes, gets fertilized, works its way down into the uterus, at which point it's a blastocyst. And the outer layer of that blastocyst are trophoblasts, which will stick or adhere to the wall of the uterus, which is called the endometrium. The trophoblasts then start to, when they adhere to the endometrial wall of the uterus, they start to multiply, to proliferate, and they form two layers. They form a layer of cells called the cytotrophoblast, and another called the syncytiotrophoblast. Syncytio comes from syncytium, a fusion of cells. Those cells in the syncytiotrophoblast will lose their outer membrane and fuse together and invade the endometrium of the uterine wall. The syncytiotrophoblasts retain their cell membrane, and they look like cells. You'll see it here. So here's the cell, the blastocyst starting to implant. It adheres. These are the trophoblast cells all around here on the outside. And there's the inner cell mass. that's going to go on to form the embryo. Here you have the endometrium, and the endometrium, the wall of the uterus, is covered with a epithelium. It's a simple epithelium, as you can see. So you have this binding of the blastocyst to the wall, and then trophoblast cells form into cytotrophoblast and syncytiotrophoblasts. And you can see the syncytiotrophoblasts are fusion products, so they're multinucleated and they penetrate into the wall of the endometrium. Now, when you look at all of the eggs that are fertilized, in other words, that are zygotes that will form following sex and fusion of the sperm with the egg, 2/3, 66%, fail to implant. So they fail to implant by the end of the first week. Remember, implantation starts at 6 to 7 days. So you have here day 6, for example. Implantation starts usually at 6 to 7 days. But 2/3 fail to implant, and therefore they don't succeed in prolonging or continuing a pregnancy. And in fact, at this stage, women aren't aware at all that they are pregnant and had a fertilized egg inside them. Among the 2/3 also, you get some that start to implant and then spontaneously abort. Among the ones that implant, about 30% of those will later miscarry, either due to genetic defect or some sort of malformation of the uterus, the placenta, or some other problem. So it's only a minority. So we're talking about 30% of the remaining, right? If 2/3 fail to implant, then we're talking about the remaining 33%. Of those, 30% will miscarry. So now you're down to about 23%. So only roughly 23% of fertilized eggs eventually will go on to a full pregnancy, roughly, and there's some plus or minus in there. Okay, so implantation involves actual penetration into the wall of the endometrium, and the blastocyst becomes completely covered with endometrial cells. So it's fully into the wall and is typically completed by about 11 to 12, average is 10 days after ovulation, which was day 14 of the uterine cycle, of the ovarian cycle. Now, one of the very first changes that can be detected, and this is the basis, in fact, of pregnancy tests, which are involve using a monoclonal antibody directed at this particular hormone. One of the very first changes is production of a hormone called human chorionic or chorionogonadotropin, HCG for short. The acronym is HCG. So it's produced and released by the pre-embryo. All right. HCG can be detected 6 to 12 days after ovulation by very sensitive antibody-mediated test involving a urine sample. The HCG is secreted by the syncytiotrophoblasts, those cells that originated from the trophoblast cells of the blastocyst and that are penetrating into the wall of the endometrium during the implantation. So the HCG, what that does is it has an effect on the, among other things, it has an effect on the corpus luteum. Remember, the corpus luteum is made up of the cells, the granulosa cells that were around the follicle in the ovary that had released the egg, the mature egg. So the mature egg came from that particular follicle, and those cells that remain behind, the granulosa cells that remain behind, become the corpus luteum, and that is what then continues to secrete progesterone and estrogen, which is very important to maintain the pregnancy, and that's due to the effect of HCG on the corpus luteum. Now those syncytial trophoblasts that penetrate finger-like extensions of fused trophoblast cells that penetrate into that are penetrating deeper into the endometrial wall go on to form what's called the chorion, and the chorion will continue to produce HCG. So the syncytial trophoblasts are producing it, and then the chorion they form the chorion, and the chorion continues to produce it, to produce these two hormones, estrogen and progesterone. So HCG maintains corpus luteum viability and estrogen progesterone production. And the production will, in fact, of these two hormones will, in fact, continue for 2 to 3 months into the pregnancy, at which point the placenta takes over. So after 2 or 3 months, the placenta is producing these two hormones, and the placenta is now fully functional, acting to provide nutrients and remove waste, is fully developed. And here you see that this is 2 to 3 months. So in this period here, that's when the placenta starts taking over for estrogen and progesterone production, as opposed to having it produced by the corpus luteum. And here you can see that quite soon after fertilization occurs, HCG is produced and can be detected, in fact, by very, very sensitive pregnancy tests on urine. Many women, in fact, once that was that kind of antibody-mediated test using anti-HCG antibodies was developed, turns out that it was discovered that, in fact, many women do get pregnant and don't even realize that they are, and the pregnancy just spontaneously is terminated because of failure of implantation or some early problem with the developing zygote. Okay, so here you see that here we have a blastocyst that is implanting in this diagram, and we're looking at formation of the placenta, which is a process called placentation. And the placenta, you need to understand, is made from both embryonic and maternal tissues. And these form a very close association with each other. The blood really is kept separate. The embryonic blood and the maternal blood is kept separate, but they're very, very close to each other. The tissues, in fact, interdigitate or project into each other with these finger-like extensions. So I remind you that the outer layer of the blastocyst are trophoblast cells, and those trophoblast cells will form into the cytotrophoblast and syncytia trophoblast cells, and the syncytia trophoblast cells penetrate deeper into the endometrium wall of the woman's uterus, the endometrial wall of the uterus, and you get proliferating and penetrating structures by the syncytial trophoblasts forming like that. You also have blood cells starting to form inside the embryo, and you have the layers of the developing embryo start to be able to be distinguished from each other. Different tissues will be derived in the embryo from different layers, embryonic layers of tissue. So you have ectoderm, mesoderm, and endoderm. And those are the ones that will go on to form all the different tissue types in the embryo. Meanwhile, the syncytial trophoblasts go on to form a chorion. And the chorion projects into the endometrium as these villi. Remember, this is the same term as used for villi in the gastrointestinal tract, these finger-like projections of tissue. Except here it's a finger-like projection of embryonic-derived tissue into the endometrial wall, so that blood, maternal blood, which is here, let me change the color, which is here in this area here, all right, that's maternal blood cells all in there, are very close to the embryonic tissue so that they can transfer oxygen and CO2 and nutrients and metabolic wastes between the mother and embryo. What you have is, as I said, the embryonic cells, the syncytial trophoblasts, go on to form the chorion, and you get chorionic villi, and these will become vascularized so that they contain blood cells, and they'll extend also towards the embryo as umbilical arteries and veins well, they extend away really, not towards. And they're surrounded, separate, but very close. The chorionic villi are surrounded by maternal blood. Placentation, or development of the placenta, involves formation of here. This is when it's fully formed. What you have is this structure here, right? This disc-shaped structure. There's the umbilical cord of the embryo, and actually penetrating into the endometrial wall to form that disc-shaped placenta. What you get, in fact, during this process is, just a second. Okay, what you get is that, remember, the endometrial wall of the uterus has a basal layer, or stratum basalis. Remember, there's the stratum basalis, and then there's the stratum functionalis. And the stratum functionalis is the layer that normally, with the menstrual cycle or uterine cycle, is forming and then sloughs off and forms and sloughs off. That's the functional layer, or stratum functionalis. It is formed from that stratum basalis, or basal layer. The part of the endometrium that lies between the chorionic villi and the stratum basalis is called the decidua basalis. And here you see that decidua basalis. So we're talking about this endometrial tissue here. Okay. That's the decidua basalis. And you have, I'm sorry. So here you have the decidua basalis, and the part of the endometrium that surrounds the space of the uterus is called the decidua capsularis. All right. So this tissue is in fact endometrial or maternal, and the tissue here of the chorionic villi is embryonic. So you have these structures here, which are the villi, penetrating in, surrounded on either side by maternal tissue. So you have the decidua basalis and the decidua capsularis. The embryo is surrounded, including the placenta, is surrounded completely by the maternal tissue. The embryo itself, looking at embryo versus placenta, is the embryo is floating within that amniotic fluid, and at this early stage, and the placenta is penetrating into the endometrium. With further development, the embryo then is completely out into the space of the uterus, and the placenta is present as a dome shape underneath the decidua capsularis. Remember, the decidua capsularis is this layer here, okay, right there. Okay. So by the end of the third month, so we're talking about 12, 13 weeks roughly, placenta is fully formed, and as I said previously, it can act completely functional for nutritive, excretory, respiratory function, and is also an endocrine gland in terms of its ability to produce estrogen and progesterone. Barriers are present that prevent free flow of substances between the mother and the fetus, so it's not a free trafficking. The two are separated from each other by the chorionic villi and the endothelium of the embryonic capillaries. So there are embryonic capillaries penetrating into those chorionic villi, but it's not, and those are, and the chorionic villi is surrounded by the endometrium and maternal blood, the endometrium of the mother's uterus, and the tissue and the maternal blood, but it does not go directly through and free flow through the chorionic villi or through the endothelium of the embryonic capillaries. Placentation also involves development of the placenta in terms of production of other hormones, and they are listed here, although I'm not going to get into any detail on these. And that's the end of Unit 8A.